Hey guys, welcome to the Neuron lecture of the final exam. This is the last anatomy video I'll be making, and so yeah, let's just go over it. All right, Neuron PowerPoint. So before I begin, I want to give you guys like a brief representation or visual representation as to what I'll be talking about. So we first have the central nervous system. The central nervous system only consists of the brain and the spinal cord, whereas the peripheral nervous system is everything else but the brain and the spinal cord. So let me see. Let me think of a good analogy to explain what the CNS and the PNS means, and explaining the afferent and the efferent divisions. So let's say you're cold, right? Now, your body telling you that you're cold is the afferent division. That's because it's going to the brain, letting your body know like you're cold. So then let's say you wear a sweater because you're cold. That's part of the efferent because you're telling your muscles to move in order to wear a sweater to keep yourself warm, right? So that's the afferent and the efferent. Just think of the afferent as information being going to the brain, and then the efferent going away from the brain. With the efferent, it also divides into two different divisions. We first have the somatic nervous system and the autonomic nervous system. And then the autonomic nervous system again splits into two more divisions, which is the parasympathetic and the sympathetic division. And I'll be going more information. Oops, what the? I'll be explaining more information throughout the video, and so yeah, let's just keep going. All right, so neurons. The neuron is the functional unit of the nervous system. It has the capacity to produce action potentials, and it has a cell body. And the cell body is a single nucleus with prominent nucleolus. The neurofilament gives the cell shape and support, and it has microtubules that move material inside the cell. The cell processes is the dendrites and the axons. So dendrites. The dendrites conduct impulses towards the cell body. It is typically short and highly branched and unmyelinated. Its surfaces specialized for contact with other neurons. So right here is the, hold on, let me erase that real quick. So this right here is the cell body, and the things branching off the cell body, the nerve cell body, is the dendrites. And remember, it's conducting impulses towards the cell body. Okay, axons. So axons conduct impulses away from the cell body. It is long, thin, cylindrical processes of cell. It arises at the axon hillock, and impulses arise from the initial segment or the trigger zone. Axons also have side branches, also known as collaterals, that end in fine processes called axon terminal. The swollen tips called synaptic end bulbs contain vesicles filled with neurotransmitters. So this right here is the, hold on, let me use a different color. This right here is the axon hillock, and this right here is the initial segment. So basically the axon hillock and the initial segment are basically in the same spot because the whole axon structure goes around like this. It says here there's side branches or the collaterals that end in fine processes called axon terminals. So the axon goes through like that, and once it splits into different branches like this, that is called the axon terminals. And it says the swollen tips called synaptic end bulb contain vesicle filled with neurotransmitters. So these little balls by the end of the axon terminals are filled with neurotransmitters. All right, give me a second, guys. So let me see where I ended off. Okay, so I ended off here. All right, continuing on, we then have the chemical synapses. So I'll give you guys the dictionary definition as to what a synapse is. A synapse is a junction between two nerve cells consisting of a minute gap across which impulses pass by the fusion of a neurotransmitter. So this right here is, let me identify the parts first. So if you recall, this is the axon terminal, and then this little bulb right here is the end of the axon terminal. Let me see if I can go back and remember what it was called. There we go, the synaptic end bulb. And in the synaptic end bulb, they're filled with neurotransmitters right here. So if you look back at the definition of synapse, it's a junction between two nerve cells. So this right here is another nerve cell, and what happens is in the junction, they pass through different informations. That's what I'm assuming because that's what it looks like in the picture. And so yeah, I really can't explain much because I kind of like briefly went over this when I was studying it, and so yeah. So at a chemical synapse, the most common type of synapse, the depolarization of an action potential reaching the presynaptic terminal causes a release of neurotransmitters which act on specialized receptors located in the cell membrane of the postsynaptic neuron. The structure and the function of the chemical synapse make them slower than electrical synapses and permit signaling in only one direction. So there's two types of synapses. The one we just went over is the chemical synapse. It is the release of neurotransmitters into a synaptic cleft, and that's why it says sound familiar because we just went over it. Now the rare one is the electrical. What happens is the cells electrically coupled as the cytoplasms of adjacent neurons are connected. It allows synchronization of neurons. And so yeah, you just know the basics of the electrical and chemical synapses. All right, going over the neuroglia of the central nervous system. A neuroglia, also called glia cells, or glia, are non-neuronal cells that function primarily to support the neurons. We have the astrocyte, microglia, ependymal, and oligodendrocytes. Going over astrocyte first. what the astrocyte does is it connects neurons to capillaries, guide migration of neurons, help determine capillary permeability, absorb K+, recycle and recapture neurotransmitters. So that's the function of astrocytes. Okay, let the train pass by real quick. Give me a second, guys. Alright, should be gone. Alright, microglia. So for microglia, it is a specialized macrophage of the central nervous system that remove the bacteria and debris around the neurons. If you look to the picture of the right, that's what it looks like, and they all float around the central nervous system. Okay, that's microglia. So for ependymal, it is ciliated, or as you can see right here, they're ciliated, and these line the cavities that circulate in the cerebrospinal fluid around the central nervous system. We're gonna go more in depth of the CSF in a bit, and yeah, but just know that these line the cavities in the central spinal fluid around the central nervous system. And then finally, we have the oligodendrocyte. What the oligodendrocyte is that it wraps cytoplasmic extensions around the neuron fibers that creates myelin sheath. So in the blue outline, that right there is the axon, and surrounding the axon are myelin sheath, and that's what the oligodendrocyte does. They wrap cytoplasmic extension around neuron fibers. As you can see right here, it's already demonstrating it. They wrap around the neuron. All right, that should be it with the 4 neuroglia. All right, protection and coverings. So in the central nervous system, you have 4 protection. You have bone and meninges protecting it. If that makes sense. We have the bone. So cranium means the skull, and the vertebral column means your spine. So the bones of those protects the central nervous system. It also has 3 connective tissues located between the bone and soft tissues of the nervous system that protects it. So it's meninges basically. Now the outermost layer of the connective tissue is called the dura mater. It is attached to periosteum, and there's many blood vessels and nerves. Now, if you recall, during the lecture, she said that there's no blood flowing into the nervous system. So, you might want to outcast this dura mater when taking the exam. Now, in the middle layer, which is also known as the spider web, it's called the arachnoid mater. There's no blood vessels. Okay. And then finally, we have pia mater, which is the inner layer. It is a nearly transparent membrane that contains nerve and blood vessels to nourish the cells. It follows contours of brain and spinal cord. All right. So comparison of structural classes of neurons. We have the multipolar, which is right here, multipolar, bipolar, and the unipolar. Now, the multipolar is the most abundant in the body. It is the major neuron type of the central nervous system. And the bipolar is very rare. They are found in specially sensory organs, such as the olfactory, mucosa, and the eye. And the unipolar, right here, is found mainly in the peripheral nervous system. It is most commonly in the dorsal root ganglia of the spinal cord and sensory ganglia of cranial nerves. And so, yeah. All right, multipolar. Right here, multipolar. Some multipolar neurons are motor neurons that conduct impulses along the efferent pathways from the central nervous system to the effector, which is the muscle/gland. Some multipolar neurons are higher level sensory neurons that convey sensory input from the first order sensory neurons to the higher central nervous levels. Most multipolar neurons are the interneurons that conduct impulses within the central nervous system. It may be one of the chain of a central nervous system neuron or a single neuron connecting sensory and motor neurons. It is essentially all bipolar neurons are, so essentially what this is saying is that all bipolar neurons are sensory neurons that are located in the special cells of sense organs. And then the unipolar, the retina are involved, the unipolar is the most unipolar neurons are sensory neurons. So most of them are sensory that conduct impulses along the afferent pathways to the central nervous system for interpretation. These sensory neurons are primary or first order sensory neurons. Okay. And so, yeah, I just basically read over it and, yep. All right. Tissue organization of the central nervous system. As you can see, right here is the central cavity. Oh, give me a second. Central cavity. And then right here is the inner gray matter. Outside here is the outer white matter. And then the gray right here is the cortex of the gray matter. But that's in the brain. For the brainstem, it's kind of the same. As you can see, central cavity, the inner gray matter, and then the outer white matter. And like 1/8 inch of gray matter in the brainstem. And then finally, in the spinal cord, the central cavity is small, and then the outer white matter is now backwards, as you can see, because right here is the inner gray matter, and then right here is the outer white matter. They kind of flip once you get to the spinal cord. I'll just keep that. All right. Myelination in the central nervous system. So the oligodendrocytes, as you know, I've previously went over, myelinate axons in the central nervous system. It is broad, flat cell processes that wraps around CNS axons, but the cell bodies do not surround the axons. So no neural lemma is formed. Therefore, little regrowth after injury is possible due to the lack of distinct tube or neural lemma. All right, myelination in the central nervous system. So white matter is myelinated processes, and of course, it's white in color. Now, gray matter is nerve cell bodies, dendrites, axon terminals, bundles of unmyelinated axons, and neuroglia, and they're gray in color. So white is myelinated, gray is unmyelinated. All right, let me see. In the spinal cord, the gray matter forms an H-shaped inner core surrounded by white matter, and then in the brain, a thin shell of gray matter covers the surface and is found in clusters called nuclei inside the central nervous system. All right, gray and white matter, that's what it's basically saying. Okay. Right here in the spinal cord, the white right here, that right there is myelinated. Whereas, in the brain, it's kind of backwards because now the white is the inner portion. And in the white portion right here, I'm not going to cover all of it, that right there is myelinated. All right, I know you guys noticed the cut here. I realized I messed up something. So I fixed it now, but yeah, let's continue. So diffuse modulatory system. So the axonal system with highly divergent projections to multiple areas of the CNS providing extensive spatial control. Each system prolongs its action by using G proteins coupling receptors. Further coverage by the release of neurotransmitter molecules into the extracellular fluid so that they can diffuse to many neurons rather than being restricted to the immediate vicinity of synaptic cleft. No idea what happened here, but yeah, let's continue. All right, going over the brain now. We have to distinct the lobes of the brain. So basically the front portion of the brain right here, that right there is the frontal lobe. And then like in the middle, right here, that's the parietal lobe. And then at the back of the brain, it's the occipital lobe. And then like, kind of like of the side of the brain, that's the temporal lobe. And each lobe has its own function. And so yeah, I can't waste any time. Let's just keep going. So the brain and neurotransmitters. So this is what the pathways of neurotransmitters would look like during different neurotransmitters occurring, right? So I don't even know what this is, bro. But I think we don't have to know all of them. We just have to know like what the basis does. So dopamine, we're familiar to that. So what happens is it starts off in the substantia nigra, and then it goes like that. All that, it just spreads around in the brain, And so yeah. All right, I'll let you guys read over this just in case, because yeah. All right, ventricles of the brain. So ventricles of the brain are continuous within each other and with the central canal of the spinal cord. So it starts off in the lateral ventricles, third ventricle, and then to the fourth ventricle. And in each ventricle, they're filled with cerebrospinal fluid. And it is lined with ependymal types of cell of neuroglia. So as you can see, right here is the lateral ventricles. Right here is the third ventricle, and then this is the cerebral aqueduct. And then here, that goes to the cerebellum, is called the fourth ventricle. And then right here's the central canal. And so yeah. So cerebrospinal fluid. So cerebrospinal fluid, or CSF, is found in and around the brain and spinal cord. It forms a liquid cushion that gives buoyancy to the central nervous system organs. CNS, yeah. It reduces the brain weight by 97% and prevents the brain from crushing under its own weight. It helps to nourish the brain as well. CSF may carry chemical signals like hormones and sleep and appetite-inducing molecules. It is similar in composition to blood plasma. It is produced by the choroid plexus that hangs from the roof of each ventricle, and it is kept circulating ependymal cells of the ventricles. All right. Motor and sensory, as you can see, we have to know this, but this is going to be in the online portion, if I recall her saying that. All right, neuronal circuits. A neuron in the central nervous systems are organized into neuronal networks. A neuronal network may contain thousands or even millions of neurons. Neuronal circuits are involved in many important activities such as breathing, short-term memory, waking up. So here are the different types of neuronal circuits. We first have the diverging. Now, diverging is single cell that stimulates many others. As you can see, single cell stimulating many others. That's diverging. For converging, it's one cell stimulated by many others. So this is the one cell being stimulated by many others. And that right there is converging. All right. Reverberating. Impulses from later cells repeatedly stimulate early cells in the circuit, which is basically short-term memory. So as you can see, I really can't explain this, but I'll try my best. It's later cells repeatedly stimulate early cells. I have no idea how to explain this, but yeah. Moving on. Parallel after discharge. Single cell stimulates a group of cells that all stimulate a common postsynaptic cell. Okay. Single cell that stimulates a group of cells right here, that all stimulate a common postsynaptic cell. This is the postsynaptic cell. And that right there is parallel after discharge circuit. Nervous system function. Starts off in the sensory input, it is integrated, and now we work it out in our motor output. So what I'm assuming this picture says is like, your body telling you you're thirsty, therefore you tell your brain to move your muscle, right here, you tell your brain to move your muscle to grab water and drink. Okay. Spinal cord. So white matter is divided into columns of myelinated axons of motor and sensory neurons. Columns contain bundles of nerve axons that have a common function or origin or destination that may carry similar information. Sensory ascends and motor descend. And it makes sense, bro. It makes sense. Gray matter, shaped like letter H, consists primarily of cell bodies of neurons and neuroglia and unmyelinated axons and dendrites of association motor neurons. So gray matter is associated with motor neurons because it's unmyelinated. I think that's what it says. Spinal cord. Roots are the 2 points of the attachment that connect each spinal nerve to the segment of the spinal cord. So the dorsal root or the posterior is sensory, whereas the ventral or the anterior is motor. So this is how I remember it. I remember PS or AM. I just need to remember one of them and then it'll tell me, because anterior is motor, posterior is sensory. That's how I remember it. It conducts impulses going back to dorsal root, it conducts impulses from the periphery into the spinal cord. Posterior root ganglion contains cell bodies of sensory neurons. Ventral or anterior root is motor, conducts impulses from the spinal cord to the periphery. Cell bodies of motor neurons are located in the gray matter of cord. So the unmyelinated of the gray matter of cord is motor neurons. Ooh, what happened here? All right, cell processes. So the bundles of neurons processes are termed tracts in the central nervous system and nerves in the peripheral nervous system. So bundles of neurons processes are termed tracts in the central nervous system and nerves in the peripheral nervous system. There's two types. We went over this, which is the axon and dendrites. But going over this again, the axon beginning at the hillock, it forms the nerve fiber and generates and conducts impulses on the axolemma. Also has secretory component at the end, which is the little ball at the end, the axon terminal, and then to the ball, which has neurotransmitters. Dendrites. It's shortly diffuse branch input. It conveys electrical information toward the cell body. Okay. All right. Spinal thalamic pathways. The lateral spinal thalamic tract carries pain and temperature. Where is the lateral? Oh, right here's the lateral. As you can see, let me use a different color. Right here's the lateral, and it carries pain and temperature. For the anterior, it carries tickle, itch, crude, touch, and pressure. So right here's the anterior, and it carries tickle, itch, crude, and touch and pressure. So the first cell body in the dorsal root ganglion with synapses and cord. Where's the first root? Right here. You can see it starts off right there. The second cell body in the gray matter of cord sends fibers to the other side of cord and up through the white matter to synapse in the thalamus. Okay, so we start here and sends it to the thalamus. Where's the second? I really can't explain this because I'm really going over this with you guys too, man. Just relax. But yeah. And then finally, the third cell body in thalamus projects cerebral cortex. Okay, that sounds pretty cool. I think I got this now. So posterior column medial lemniscus pathways of the central nervous system. Proprioception, vibration, discriminative touch, weight discrimination, and stereognosis. Signals travel up the spinal cord in posterior column, and fibers cross over in medulla to become the medial lemniscus pathways ending in thalamus. The thalamic fibers reach cortex. Details of motor pathways. So the lateral corticospinal tract, its cortex, cerebral peduncles, 90% decussation of axon and medulla, tract form in the lateral column. It's associated with skilled movement in hands and feet. For anterior corticospinal tract, the 10% axons that do not cross, it basically controls the neck and trunk muscles. All right, and then the corticobulbar tracts, it's cortex to the nuclei of the central nervous system. 3, 4, 5, 6, 7, 8, 9, 10, and 11. But they're in what's it called? They're in Roman numeral. So Roman numeral 3, 4, 5, and all that. And it is associated with movement of eyes, tongue, chewing, expression, and speech. All right. The peripheral nervous system. We first have the sensory. It is in the afferent division to the central nervous system via the somatic and visceral afferent fiber motors. Afferent fibers, sorry. Now the motor is efferent division from the central nervous system to the organ, muscles, and glands. And then the motor splits again to the somatic and the autonomic. Going over the somatic, it's the voluntary nervous system from the CNS to the skeletal muscles. So the voluntary for somatic. And the autonomic is the involuntary nervous system, including the smooth muscles, cardiac muscles, and glands. All right. Now dividing more into the autonomic, we have the parasympathetic and the sympathetic. Going over parasympathetic, the parasympathetic division, which innervates viscera in the body cavities, plus genitalia and hand viscera, functions to restore routine visceral status and implement specific visceral functions. So think of parasympathetic as going back to homeostasis. The sympathetic division, which innervates viscera throughout the body, also has the routine function, but its most dramatic role is to prepare for energy-intensive fight or flight by mobilizing a global visceral response. So the word fight or flight should give you the green light as to what a sympathetic is. And so yeah, going over more over the parasympathetic and sympathetic. The parasympathetic constricts pupil, whereas the sympathetic dilates pupil. Parasympathetic stimulates saliva, whereas the sympathetic inhibits salivation. Oops, sorry. Parasympathetic slows heartbeat, and the sympathetic increase heartbeat. Parasympathetic constrict airways, and then the sympathetic relaxes the airways, so you can breathe more. Parasympathetic stimulate activity of the stomach, whereas sympathetic inhibit activity in the stomach, because for sympathetic, your body wouldn't need to be digesting food when you're in the fight or flight mode, right? For parasympathetic, it stimulates gallbladder, and then sympathetic inhibits gallbladder. Parasympathetic stimulate activities in the intestines, whereas sympathetic inhibits. For parasympathetic, it constructs bladder, and then for sympathetic, it secretes epinephrine and norepinephrine. I don't even know what that means. And it also relaxes the bladder. All right. Autonomic nervous system, parasympathetic, and the sympathetic. So it looks like for parasympathetic, the preganglion is long, and then the postganglion is short. For sympathetic, the preganglion is short, but the postganglion is long. Let's see. So sympathetic is short. Yeah, that makes sense. So this is what I'm gonna write during the exam in a few hours. I'm gonna do this, and then I'm gonna write para. Oops, no, not para. Sim, and then long like this, para. So that can tell me during the exams, like, if sympathetic or parasympathetic. Now this is not one of them. If you see like a line just going like that, that right there is somatic, which is basically both. All right. If you're still lost, this is what I'm referring to. So as you can see, the no ganglion is the somatic, the short preganglion is sympathetic, and then the long preganglion is parasympathetic. So if you're still confused, that's what I'm referring to. All right. Parasympathetic and sympathetic division. Now I kind of went already over this with this slider here inside. Let's just skip it. All right, let's see what's going on here. Sympathetic trunk and pathways. So it looks like we start off from the spinal cord. Remember, information travels up and down the spinal cord like that. And so yeah, let's say you're sweating, or not you're not sweating, but let's say you're hot and you need to sweat to cool your body down. Information travels travels down the spinal cord to the motor. Remember, the motor portion is the anterior part of the spinal nerve, or the anterior root. So right here, your body tells you you need to sweat. So information travels down the spinal cord to the motor, which then goes to the effector of the skin and blood vessels. Now, some information travels up like that via the communicating rami and the sympathetic trunk, but it still goes to the same spot. It basically just depends as to where you need to sweat. Actually, you sweat everywhere. I have no idea why it goes up, but yeah. All right, moving over. All right, so we already kind of went over this. As you can see, starting off at the sympathetic, right here, sympathetic, the preganglion is short. However, the postganglion is long. And it makes a stop by in the sympathetic ganglia. Now, the sympathetic ganglia are the ganglia of the sympathetic nervous system that delivers information to the body about stress and impending danger, and are also responsible for the familiar fight or flight response. So that's sympathetic. For parasympathetic, it has a long preganglion, but short postganglion. That's all you really have to know with that. All right, the autonomic and somatic nervous system. So the autonomic, the one we're kind of doing right now, the autonomic has two neuron pathways. So as you can see, we have the preganglia right here, it makes a stop here, and then the postganglia. That's two neuron pathways. Whereas the somatic is just one neuron going through. Yep. All right, the basic components of all human reflex arcs. Here we go again with the stupid train. Give me a second, guys. I'll make a cut. All right. The basic of component of all human reflex arc. So in this picture, what happens is the nail pokes into the skin, which activates the receptor under the skin that sends information to the spinal cord. And remember, the posterior side is the sensory. So as you can see, sensory travels up in the posterior side, which is then integrated in the spinal cord. If you recall, the spinal cord and the brain are the integration center. So it integrates the information, which then tells your muscles to basically move, or not, to remove the nail, right? So then what happens is the sensory then becomes motor to the anterior side and tells your muscles to remove the nail. So that's basically what's happening here. All right, distribution of the spinal nerves and posterior view. So the cranial nerve is from C1 to C8, the thoracic is from T1 to T12, and the lumbar is L1 to L5, and the sacral is S1 to S5. And then the cochleogeal, I don't know if I said that right, it's C0. All right, referred pain. So visceral pain that is felt just deep to the skin overlying the stimulated organ or in the surface area far from the organ. Skin area and organ are served by the same segment of the spinal cord. Heart attack is felt in skin along the left arm since both are supplied by the spinal cord segment T1 to T5. Reflex activity. Reflex activity is a rapid, predictable motor response to a stimulus. It is involuntary. It occurs through reflex arc. Receptor of stimulus, sensory neuron to the central nervous system, integration center, motor neuron conducts information to the motor center, and then an effector response to the stimulus from the CNS. So I basically explained it right here with the nail. Histology of the nervous system. Supporting cells in the peripheral nervous system, which is the satellite cell and the Schwann cell. The satellite cells surround the neuron cell bodies within the ganglia, and the Schwann cell surrounds and form myelin sheath around the larger nerve fibers. It has the same functionality to oligodendrocytes. All right, regeneration and repair. So plasticity maintained throughout life. It is the sprouting of new dendrites, synthesis of new proteins, and the changes in synaptic contacts with other neurons. However, it has limited capability for regeneration. In the peripheral nervous system, repair is possible to damage dendrites or axons. However, in the central nervous system, no repair is possible. All right, now we're going over the health stuff that we kind of have to memorize according to Dodenoff. So cerebral palsy is the loss of motor control and coordination. It is the damage to motor areas of the brain, usually caused by infection of a pregnant woman with the rubella virus, radiation during fetal life, or the temporary lack of oxygen during birth. It is not progressive but irreversible. Parkinson's disease. It is a progressive disorder striking victims at age 60. Environment toxins may be the cause. Neurons from the substantia nigra do not release enough dopamine into the basal ganglia. Effects are tremor, rigidity, I don't know if I said that right, bradykinesia, which is slow movement, or hypokinesia, which is decreasing range of movement. It may affect walking, speech, or even facial expressions. And the treatments to treat Parkinson's disease is drugs that increase dopamine levels or prevent its breakdowns, surgery to transplant fetal tissue, or removal part of the globus pallidus to slow tremors. Neurogenesis in the central nervous system. It is the formation of a neurons from the stem cell that was not thought to occur in humans. In 1992, a growth factor was found that stimulates adult mice brains to multiply. In 1998, new neurons found to form within adult human hippocampus, which is area important for learning. And then the factors preventing neurogenesis in central nervous system is the inhibition by neuroglial cells, absence of growth stimulating factors, lack of neurolemmas, and rapid inflammation of scar tissues. Multiple sclerosis. It is an autoimmune disorder causing destruction of myelin sheath in the central nervous system. The sheath become plaques or scars, and it affects 1.5 million people in the US. It appears between the ages 20 and 40, and females twice as often as males. Symptoms include muscular weakness, abnormal sensation, or double vision. Remissions and relapses result in progressive cumulative loss of function. And then finally, we have epilepsy, the second most common neurological disorder that affects 1% of the population. It is characterized by short, recurrent attacks initiated by electrical discharges in the brain, which is light, noise, or smells may be sensed. Skeletal muscles may contract involuntarily, and loss of consciousness maker. Epilepsy has many causes, including brain damage at birth, metabolic disturbances, infection, toxins, vascular disturbances, head injuries, and tumors. And that is the final thing in the PowerPoint.